Hello, we're ready to start this next session. It's wonderful to see all the collegiality and discussion out there. Sorry to interrupt. The title of this next session is Regulatory Issues, Translating Research to the Clinic. And we will have some very distinguished speakers here, and I will introduce the first one. And I'll introduce myself when it's my turn. But it's now time for us to consider what we need to do to move all of this forward in a regulated way. And as you know, in the US, we have to deal with the FDA. So our first speaker is Dr. David Ball. He has decades of experience as a HCLD lab director in assisted reproductive technologies. And David is going to be our voice of reason for the things we need to consider as we move ovarian and testicular tissue cryopreservation and transplantation to clinical practice. So David.
Hello, good morning, and thank you, Mary. Yes, well, this is going to be a much more mundane talk than you've heard pretty much throughout these sessions here. And I do have to comment that this is my first Onco meeting, and I have to say the depth of the scientific investigation is truly impressive, as is the breadth of the discussions going on here. I mean, everything from the basic science to counseling of patients and so forth. It really is impressive, and I think you should all feel very good about that. But I would like to start off by saying thank you. Number one, as I said, the basic research here is nothing less than superb in my view. Meeting organization has been extremely well done. So on that point, thanks to Kyle, Mahmood, the support staff, which is large, I can't name them all, and the financial support, of course. It couldn't happen without that piece of it, and I certainly think the results and discussions of this meeting have earned that support. So, and last but certainly not least, Teresa Woodruff for having the vision years ago now to start this group working together and so forth. So all due thanks to all of those people. Okay, so quick background for me. Number one, I was trained as a basic researcher, and this dates back decades, of course, into the mid-late 70s, University of Wisconsin doing mostly in vitro maturation of eggs from cattle, okay? And of course, a lot of the work from that is spun off into other areas, including human IVF. But I've also been lab director since 1983, started the IVF program at Mayo Clinic in Minnesota. And since then, having defected from research to clinical application, for 20 plus years now, worked multiple labs around the country, and mostly in private practices, but not solely. I currently direct 3 labs, IVF, ART labs, in 3 different clinics, including UPMC here in Minnesota, or in Pennsylvania. And about a month ago joined this outfit called Fertility, which really it's a bit of a different deal. I can explain it to you if anybody's interested. But I am relatively new to this field. So in advance, I'll say I have no doubt I'm going to make some missteps here in what we try and communicate. And I still am trying to get a good feeling for the consortium structure here, how the administrative part of this actually works, okay? But just to introduce the topics I'd like to at least touch base on. We can't go into a lot of depth on these. It's going to be somewhat superficial just from time constraints, but there is a lot to all of these, which we can perhaps address at another time. But technical, things like cryopreservation, regulatory, which I think is not there too much right now, but I think the field has to prepare for, and then organizational structure, how to basically get all these groups working not just in their own silos, which is important, but also as a true group, okay? And I think the future does have some potential non-scientific challenges, which we'll try and address. And having said this, so going back, dialing the clock back to the early 80s when I got involved in IVF, for example, on a clinical level, there are some real parallels I see with where this field is at this time. So, okay, this picture, in full disclosure, I have no financial interest in Southwest, okay? But I'm using it because I kind of see this field right now as a plane taking off. There's been a lot of work on the airplane itself, taxiways and so forth, but now it's starting to really take off into full flight. Okay, so let's go into this a little bit. So technical considerations, and I will address primarily ovarian tissue, not to dismiss by any means testicular tissue, but due to, again, time limitations, I think I'll stick to this, understanding that there are some differences, but in general, I think some of the concepts are the same, okay? So it's clear to me the most common method, certainly at this point, for freezing these tissues is with slow cooling. I was surprised to learn this, quite honestly, because we were doing this in IVF, even in cattle and certainly in humans, back in the early 80s. I mean, this is an old, although proven technology. And in the IVF field, this has been almost totally supplanted by vitrification, okay? Certainly for eggs, gametes in general, and certainly for embryos, it's a much superior technology. I'd be surprised if at some point in the future, this doesn't take place in these tissues we're talking about here as well. Mary Zielinski's done some beautiful work in this area, and she can address that much better than I. And there are hints of vitrification in the literature even, as I'm sure you know. But at this point, I'd say it's somewhat, well, it's not been proven on a wide scale, right? I've even had interest in vitrification on a more clinical level, even these microsurgical retrievals of sperm samples and so forth, to actually freeze these very few sperm in a large vial of medium. Doesn't make a lot of sense to me. If we could do it in much smaller volumes on cryotops, something like this, I think it would be much more useful clinically. But anyway, the point here, vitrification does need to be further development, of course, to take it beyond proof of concept to actual proof that it works clinically, okay? All right, now let's drift over into the regulatory concerns, okay? So processing and freeze media, there are very many variations on the theme, if you will, that I see in the literature. One that concerns me a little bit is I do see some reference to fetal calf serum, and I do question if that's ultimately a good idea, particularly when we put this in the realm of regulatory control of the field, okay? And while I'll address specifically U.S. issues with federal or FDA, I know I'm not excluding European because I know they have their own regulatory sets. I'm just not familiar with them. But I do know from the human IVF field, using proteins from another species, FDA will have concerns about that, okay? Overall, FDA does consider the freeze media as a device, okay? Which basically opens the door for them to come in and start to impose regulatory limits. Okay, so we have to be aware of that. And looking forward, this is, I think, potentially going to be a concern. Currently, just so you know, there are no currently cleared FDA cryopreservation for ovarian tissue on the market. Okay, so this brings up other issues, which I'll talk about in a minute. But then the other part of this that I think is an issue at this point is ASRM, as you all know, has basically removed the experimental designation for ovarian tissue cryopreservation. Honestly, I'm not sure that was wise. I'm not sure it was helpful for the field. I mean, I know in the US, for example, there's very little clinical demonstration that this process actually works in terms of production of live babies, okay? So in my view, largely it still is experimental. And just based on the work I've seen here presented the last couple of days, I think that's fitting, okay? And then the other part of that ASRM stance is, will these regulatory agencies like FDA honor that stance? And quite honestly, in my view, I think the answer is questionable. I'm not sure they're going to go along with any professional organization statement on this. They have their own rules, okay? So, to back up on this, how I got involved in this, and this was one of these situations I wish I'd have never asked this question, okay? But we were ramping up the ovarian tissue cryo project in Seattle, which is where I just left a month and a half ago. And so trying to prepare for that from the laboratory standpoint, okay, we need to get medium in stock and so forth for this. And at that time, Origio was the only one making this commercially, okay? And my understanding is they had a contract with the consortium to do this, in essence, on an experimental basis. Origio had some interactions with FDA that made this okay to do, all right? Well, when I went to start ordering medium, I saw it was no longer on their website as an option to choose to order. So I wound up contacting Origio, and they said, well, we're not sure we're going to be making this anymore, okay? And of course, your antennas go up immediately because in my view, again, dialing this back to the 80s in the IVF world, we're in the laboratory, we're making all this medium up. Quality control consisted basically of, is the pH right? Is the osmolality right? There's really no other contamination ability to test this medium and so forth. So my concern is if we have no commercially available source for this, which has standard manufacturing processes, QC and so forth, that's going to be a problem for an IVF lab. Okay, so let's assume for a moment Origio does not do this, does not continue to make these media. Okay, now what, right? Well, the only way I see it happening is going back to the individual clinics that are performing these procedures. And as I mentioned, this raises a whole host of problems, I think. And then even from a regulatory standpoint, freezing, thawing these tissues for auto-transplant are the beautiful work I've seen here, even on in vitro development of follicles in the laboratory. Will these tissues ever be used for donation to another patient, whether it's a sister or a relative or anonymously? For sure, FDA is going to have interest in that, okay? For those of you involved in IVF labs, for example, anybody doing anonymous donation, there's a whole list of things you have to do to qualify a tissue for that use clinically. Okay? So as the field moves from research, and again, I think that's where I see this field right now, it's trying to transition appropriately from really research arena to clinical application on a wide basis, okay? And that's going to bring some different things than probably most people have thought about so far, and I think that's part of the reason for introducing this at this talk, okay? And just so you know, I think right now this field is kind of flying under the radar, so you've probably not heard from these regulatory agencies, but it's not going to take too much notoriety, whether it's a lawsuit or a bad news article from an experience a patient had with this, to all of a sudden it's now on their radar and they're going to become interested. So this is looking forward here, okay, trying to be ahead of that curve, if you will. And just so you know, FDA does consider, or looking at the regulatory agencies, what would be their interest? So FDA looks at products, okay, basically quality of products. CDC, their concern is patient safety, which ties together, to me at least, with product, okay, quality product. And then there's all sorts of health agencies at state level, and including CLIA, if you're going to do any kind of diagnostics on this at a clinical level, they're going to have interest in this most likely, okay? So let's move to operational considerations for a moment. So if a commercially made, quality-controlled media is not available, who will make it? Well, right now, a lot of these tissues are being frozen in IVF labs, and I've got some more comments on that. But if it is in an IVF lab, number one, so from a practical level, every IVF lab in the country right now is swamped, just taking care of infertility patients straight up, okay? So the ability or the capacity to now treat this extra whole different group of patients, I think is a practical issue. It may not be there. And then, of course, just again, from a practical standpoint, as you well know, certainly better than I, some of these tissues need to be processed on relatively short notice, okay? And again, if this tissue winds up in your lab after a full day of embryology work at 5 in the afternoon, that's going to be tough to staff for that. People just aren't, number one, trained for this in most cases, and certainly everybody has a life, so staying in the lab 12 hours a day is going to be tough, okay? Which media constructs should be used? I don't think we know that yet. You know, looking around at the literature, there are a lot of variations on this theme. I have some concerns, quite honestly, with some of the buffers being used, but won't get into that now. And then even if it's made in an IVF lab, how do you QC it? You know, we simply don't have the capacity to do, you know, bacteriological work and this sort of thing to actually QC these on any kind of uniform standard. And then ultimately, one of the concerns becomes, at some point, liability, okay? And my point being this. So take the example of a patient goes through ovarian tissue cryo, ultimately has a transplant and comes down with an infection, okay? We all know in the US, it's a litigious society, right? So somebody's gonna get sued for that, okay? And if I were the lawyers, thank God I'm not, but if I were the lawyers, I'd say, okay, let's go back and see who touched this at any point in this. And if now it's in the laboratory, you're using media that you constructed in that facility, and you had minimal or no QC on it, they're gonna be all over you. So I think we gotta be really mindful of that possibility and trying to avoid it at all costs. To me, this comes back to ideally a commercially prepared product, okay? All right, so my big question, again, coming from the viewpoint I do, that is IVF labs on a clinical level, are IVF labs the best place to be freezing these tissues? In my view, this is macroscopic work for the most part, okay? The one place IVF lab may be useful is with freezing eggs. We do thousands of those, okay? It's a well-structured and a routine procedure for an IVF lab. But for ovarian tissues, again, to me, that's macroscopic work, not microscopic work. Most patients don't register with an IVF clinic. They're not true infertility patients, if you will. They're not there to become pregnant as soon as possible. So tracking these patients down the road, the infertility clinic, I don't think can do that. You know, the tissues may come from a hospital down the street, we freeze them, send them off to a long-term storage facility. The patient may never use them, or they may use them in a different city or state altogether. And there's no way for an IVF clinic to really track that, at least as it's structured now. Let's see. So if not an ART lab, where? Well, I think there's several possibilities here, probably none of them great, but in my view, probably better than an IVF lab. But if you think about this, the tissues, of course, are going to be removed in a surgical suite of some kind, right? Most likely in a hospital, I would guess. And then now to transport that, and of course, Kyle's model right now is to ship it across country in some cases and so forth. And by the way, one question I don't have a good sense of is what transport time is acceptable. Honestly, I don't know. My thought is probably the less time, the better. If that's true, then perhaps processing right at the site of collection in the hospital is the best. There's really no transport time, perhaps down a hall or to a pathology lab somewhere close by at least. So it would eliminate any transport issues that might exist. And then perhaps another model, and I think the Europeans have this and seems to work quite well for them, is regional centers to process these and freeze these tissues. I don't know how many it would need, but as you know, U.S. is a big country, so I can see the possibility of several of these being available and even have to plan for backup, you know, weather conditions, you know, flights across the country, this sort of thing could be a problem. So you have to be relatively light on your feet to change at the last minute to which center this may be going to. But even beyond that, then we look at competency assessment. Who should be freezing these tissues? You can't be the latest person of the day in the lab that can't say no, right? So there needs to be some real training. They have to know what they're doing. And then again, if we don't do this, that raises the liability issue again to me, okay? So this is a far out one, okay? But at least in theory, it's a possibility. Should there be trained technicians basically in a staff somewhere ready to go on a relatively short notice to travel to the site that these tissues were collected? That does raise a whole lot of other practical issues, but in theory, at least, that might be an answer. Okay, maybe all of these, okay? Okay, so future considerations, and I've heard some superb talks about this. In fact, the last one we just heard. I think this field is really ready for a true registry. All the data, in my view, in an ideal world, would be sent to one center, so every patient that undergoes these procedures is in a database. You can track them potentially, collect data, which is going to be critical for this field. And again, as a parallel to IVF, we are required to report every single treatment outcome to Centers for Disease Control. Okay, I wouldn't be surprised at some point in the future where they're concerned for patient safety, that becomes an issue for this field. Okay, so being able to track these outcomes, you're going to need a centralized database, in my view, to really have accurate reporting. Okay, so lab, where they were used, or where they were frozen, procedures used, all of this business. We saw some very nice, I think, in essence, demographic and procedural survey questions in that last talk just a moment ago. Okay, so who's best positioned to manage a registry? Number one, should there be minimal qualifications even for ovarian tissue cryo or testicular cryo for that matter? And this would include clinic facilities, lab equipment, and surgical suites that do the procedures. The personnel qualifications. This is routine for ART clinics. You have to have qualified people documented with training and so forth and experience to actually be able to perform these procedures. So training, documented training, routine competency assessment of the people doing the work. The clinical staff, are they qualified to even counsel patients? The lab staff, of course, to actually do the procedures. And then one of the things I was impressed with, because honestly I had not thought of this, but even extending out to counseling of the patient's wellness of the patient to making sure they're well-informed and comfortable moving ahead with these procedures. So future regulatory possibilities, I think, that are on the horizon. Number one, I think the field should anticipate government and other agencies becoming involved with regulatory oversight. I got to believe as soon as this fully transition into clinical practice, this will happen, okay? And I think probably the best way to deal with that is to have an organization like this with a clear structure that will benefit any compliance dictates, okay? So it has to be really well organized. I see some beautiful work being done, but I think some of it, understandably, is in silos to some degree, but I think the leadership needs to get their arms around this and pull it in tight. Okay, so technical considerations, IVF, in vitro development of competent gametes, that I think will be a game changer. And again, I've seen some beautiful work presented here. But then that raises whole other issues. Where should that be performed? That's not IVF lab work in my view. This is pre-IVF lab work, okay? So I think this would take probably different facilities, different staff, certainly different training, and so forth, different qualifications of the people doing it. And then it in itself will probably face regulatory issues as well, for the same reasons we just talked about: competency assessment, quality control, these sorts of things. Okay. What quality control will be needed? Those are questions that have to be answered. What must be proven to move these procedures from research to a clinical lab for true application on a widespread basis? Again, regulatory bodies will undoubtedly show a presence once this comes on their radar and they know this is being used in patients. Okay, future considerations, close to the end here. expect there will be research material available to this group, right? And so then those questions become how many, or many with high quality work initiatives and hypotheses will likely raise their hand, hey, I need some of that tissue. So how does that tissue then get distributed? Well, it seems to me you probably need to set up some kind of a research committee and in essence have those that are asking for the tissue create some kind of proposal that the committee can review, and then in essence prioritize what seems to be the best constructed studies and probably or possibly the most important impact on the field to actually distribute these tissues to. Okay? So in summary, this field, in my view at least, is in transition from research and development to potentially widespread clinical application. Okay? The leadership, I think, of this group really does need to be looking to come challenges and prepare for that. Some of these I've tried to describe just in the last few slides. Okay, so 4 key points, I guess I'd say. Number one, in my view, it's critical to have a commercially available freeze medium to be doing this with. Otherwise, honestly, I think it's going to be chaos and leads to potential liability concerns, and mostly from QC standpoint. Okay, unified registry, again, we just saw a beautiful presentation on that. I understand that's in one center right now, but I think this, as much as possible, needs to be uniform across every group doing this. Okay, database is critical for outcome analysis. Okay, anticipate regulatory agency involvement. They will be looking at this at some point, okay? And then a clearly structured administrative arm to coordinate the efforts on research, clinical usage, and outcomes in response to regulatory agencies' interests, okay? So last point, thank you very much. My first meeting here, or first attendance at this meeting, it's super impressive, folks. You really ought to feel good about this. Okay.
Thank you.
Thank you for not throwing anything, okay?
Stay there. We have time for a few questions. Thank you, David, very much.
Hi, I'm Monica Laranda from Lurie Children's. We do some ovarian tissue cryopreservation processing at our site. And so I just wanted to reiterate one of the comments that you had made, that I agree that it would be great to have some quality control measures, and I've talked with Kyle about this too, before we get told kind of what to do. I think that we should really, by FDA and CDC or other or CLIA or other regulatory bodies, that I think we really should take notes from what our European colleagues have done, and quality control by xenografting and things like that, to ensure that the processing and the technique is appropriate for and successful with transplant. But I think that this is one of the greatest forums that we could probably come together and do that, like Leslie is doing in Colorado with the database.
Yeah.
It's just a comment, and I would think that this is a great opportunity
Totally agree.
group
Yeah.
to do this. Yeah.
Well, I'm not sure I heard the first part of that, but if your comment was to reach out to FDA and CDC for their input, I would not do that.
Yes, thank you so much. I'm Erin Rowell from Lurie Children's also. I work together with Monica, and we opened our own gonadal tissue processing suite in December 2020 in the Children's Hospital operating room sterile core right across from the OR where we remove tissues, because we were having some of the exact issues you described, which is that as a children's hospital, even within Northwestern, where Teresa has been and where we had great buy-in for processing ovarian tissue, we found that it was really hard for the adult lab to be as nimble and flexible as we needed them to be with pediatric patients who are being diagnosed really quickly, need to get to the OR within a day or two, not be anesthetized multiple times, and also we needed the opportunity to be able to do more than one in one day for OR time if we needed that. And so we have found that by having our trained team to do that. And the other piece of that I would say is the prepubertal patients. We've really in the past year and a half discovered a lot of important things about how we're processing tissue for prepubertal patients in terms of what is quality, how many strips of tissue is enough, how do you do the thinning and scraping in a way that's appropriate for prepubertal ovary. And some of that work was presented here as a poster and will be coming out. But I think, again, it really highlights that those of us that particularly do pediatric OTC, where that is the only pretreatment option for some of those children, we really have the opportunity to harness the power of this group to do that. That's even better.
Well, thank you for that comment. I just as a bit of an aside, I mean, from my IVM work and even in the clinical application, IVF lab, we're very studious about maintaining temperature, okay? And the point being there, at least for oocytes, cooling these things off, I mean, you get microtubule disassembly, and it impacts the oocyte, okay? Now, if you put them back in warm temperature overall, they do tend to reconstruct, but I don't think anyone's looked at, okay, if it's cold at 4 degrees centigrade, if it's being shipped across the country, for example, does time matter for the ability of that oocyte to recover and reconstruct the infrastructure of the egg, if you will? So I don't know, but what it sounds like you've adopted is very much one of the proposals I'm suggesting. Do it as close as you can to where it was harvested. Yeah, so thank you.
Thank you very much.